Good evening, I'm Katunje Brendan. We are group 22. I'm going to talk about the EDA visualizations of our EDA explanatory analysis. We are going to start with the correlation heat map. The correlation heat map, first of all, it provides a comprehensive relationship between all variables in the dataset using the Coulomb color distributions. It displays the correlation coefficients ranging from -1, which is deep blue, indicating the strong negative correlation. It goes up to +1, which is deep red, which indicates the strong positive correlation. The anointed values show the exact correlation coefficients, which helps us identify the strong positive or negative relationships between the variables. It also helps to identify the independent variables that show minimal correlations. It also, lastly, helps to identify the potential multicollinearity issues that might affect further analysis. Then we are going to go to the box plot features. The box plot features helps identify the central tendency and the spread of each feature. It also helps identify the presence and extent of outliers. It identifies the asymmetry of the distributions and also the differences in scale between the features. Talk about the box plot. The visualization provides the statistical summary of each of the features distribution, showing the median, which is the central line. It also shows the interquartile range, which is in form of the box. It shows the whiskers extending to the most extreme, and then the outlier points. It shows the individual outlier points, and then helps identify the above features. We are going to take infestation versus the ECF scatter plot. This one shows that this is the scatter plot, which examines the relationship between the tick infestation levels and ECF occurrence. Each point represents individual case with the x-axis tick, which is the tick infestation level, and then the y-axis is the ECF status, which is either 1 or 0. The visualization we are seeing here, it helps us to identify whether there's a higher tick levels correlate with the ECF occurrence. Also, it helps identify the possible threshold effects in the tick numbers, and also helps identify the patterns that might inform the prevention strategies. We are going to the geographical distribution of ESF. Here we use the longitudes and latitudes. This visualization maps the spatial distribution of ECF cases using the x-axis, which has the longitude, and the y-axis, which has the latitude. The color coding is the ECF status using the warm color. It helps us to understand the geographical scattering of the ECF cases. Helps us to understand the potential environmental risk factors, and then the areas that might need targeted intervention. We are going to genotype distribution. This visualization shows the proportion of two different genotypes, which is either 1 or 0. They were formerly 54K and 800K before we did encoding of the numerical values. In our dataset, the pie chart makes it easy to compare the relative frequencies at a glance, and also that each genotype is represented by a different color with the exact counts labeled. This helps us to understand the genetic diversity in our sample population. It also helps us to make it easy to compare the genetic relative frequencies. We are going to ECF distribution. It helps us to understand the ECF prevalence in the population. And then this pie chart is in a format that makes it immediate apparent for any imbalances. As we can easily see from the structure, it is properly balanced, which prevents any biases during our training of the model. We are going to tick versus the Cape scatter plot. We are talking about the Cape buffaloes, which are the animals that spread the ticks. Among its functions of this scatter plot is that it helps us to identify any patterns or correlations between these two variables. The clusters of points indicate common combinations, and then the outliers represent unusual cases which are worth investigating. This scatter plot explores the relationship between the two counts, which is the tick and then the Cape buffaloes. The chart displays the distribution of Bio5 values across the dataset. Bio5 typically represents the maximum temperature of the warmest month, which is an important environmental variable. The bar chart format helps to visualize the frequency of different Bio5 values, which makes it easy to identify the common ranges and potential outliers in the environmental data. It helps us to visualize and identify the common ranges and where we can put a lot of investigation. We shall move on to average tick by cell. This is the line chart. It tracks the average tick count across the different cells. This line format helps to visualize the trends and patterns in tick distributions across these units. The sharp changes in the line indicate environmental or other factors affecting the tick population in particular cells. It helps us to track the average tick count across the different cells and also visualize the trends and patterns in the tick distributions. Lastly, I'm going to talk about the geographic distribution. This scatter plot helps to map the spatial distribution of our data points. Each point represents a location where the data was actually collected, which is the longitude on the x-axis and the latitude on the y-axis. This visualization helps to understand the geographical spread of our data area and also identify the spatial parts in the regions of higher concentration. Thank you.
Good evening, everyone. My name is Benam Shadek, and I'll be continuing from where my colleague has stopped. So, we shall proceed with the cattle versus the Bio5 relationship. And as we see in the slide, this visualization, it explores how cattle numbers are affected by the maximum temperature, which is the Bio5. And understanding this relationship can help determine whether there is an optimum temperature, where there are optimum temperature ranges for cattle populations, or if extreme temperatures negatively impact the herd sizes. So the temperature is the key factor here. Then we can talk about the average cattle by farm, where this line chart tracks the average number of cattle per farm and helps us understand the variations in herd sizes between different farms and whether this affects the ECF transmission. So if a farm has more cattle, this visualization will show us that is it on high risk of being attacked by East Coast fever. Then next we're going to talk about the tick distribution, as we see in the slides, where the bar chart provides the detailed view of the tick counts across the data set and the height of each bar, as we observe in the slide, represents the frequency of different tick levels, and this helps us identify areas with high tick infestations. Then next we are going to talk about the CAF versus the ECF relationship, where, as we observe in this slide, the visualization, which is a scatter plot, it examines whether the CAP characteristics correlate with the ECF presence. Now, if I'm to remind all of us that the CAP we're talking about is the Cape buffalo that spreads these brown ticks that spread the East Coast fever, and once your farm is next to game parks or wildlife reserves, your farm is on a high risk of being attacked by East Coast fever, because these ticks will move from the brown, these Cape buffaloes, to your animals to infect them. So, going back to the Cape versus the ECF relationship, if a pattern exists this could be a useful, if a pattern exists between these two comparisons, then this could be useful for predicting the disease risk based on the CAPE data. Next, we're going to talk about the Bio5 versus the ECF relationship, where this scatter plot examines the relationship between maximum temperature Bio5 and the East Coast fever cases. And if there is a temperature-dependent pattern, it could help in predicting the disease hotspots. So, if there is a it can help us if we find that there is a relationship between the temperature and the ECF, and we identify a hotspot, like maybe during this period of time, this period of this weather, there was a high risk or there was a high record of ECF cases, then that would help us to identify the hotspots and help the machine learning model easily predict the ECF cases. So next we're going to talk about the ECF rate by cell, and this line chart tracks the proportion of the East Coast fever cases across different cells, and it is valuable in identifying these hotspots where intervention efforts should be prioritized. So once a hotspot is identified for East Coast fever, it should be prioritized and the necessary control measures should be put in place. Then we can talk about the average bio5 by cell, and this chart shows how the average maximum temperature bio5 across different cells, and understanding the thermal landscape helps us to assess how temperature influences the tick populations and cattle health. Remember, these ticks, they have specific way at which they thrive, where that's why we can find that in a particular season we record many cases of East Coast fever. Then next we can talk about the cattle distribution, where this bar chart provides a clear view of cattle herd sizes across the data set and helps us understand in understanding the population density variations between different farms and regions. Last but not least, we can talk about the CAPE distribution, which is a bar chart that displays the CAPE measurements across the data set and helps identify the most common CAPE values, their variation, and any unusual patterns or clusters worth further investigation. Lastly, we're going to talk about the balanced ECF class distribution. And as we observe in the visualization, the slide, it shows how ECF cases are distributed after balancing the dataset. And ensuring the balanced dataset is very important for reducing bias in machine learning models and improving prediction accuracy. So in summary, through these visualizations, we have explored the key factors influencing East Coast fever risk, including the genetic traits, tick infestations, environmental conditions, and farm distribution. And these insights are crucial for improving disease prediction models and guiding targeted intervention strategies for cattle farmers. So that's it from us. Thank you very much.